So hopefully it isn't going to come to as a surprise to anyone, but exam 3 will be on Monday. It's going to cover chapters 11 through 16. I purposely, and we're gonna do chapter 16 today, finish it today. I purposely put a little bit more into this third exam because, at least in my opinion, it puts a little bit less on the fourth exam, and a lot of the fourth exam is going to start coinciding with all of your other exams. So I think it'll work out a little bit better this way. So what we want to do is finish chapter 16, which really just has two parts to it. All of it focused on operons. The first is to talk in a broader sense about how operons are regulated, and then the second to tell you everything you've ever wanted to know about the Lac Operon. And hopefully it will benefit you in other courses like micro and biochemistry, since it is one of those topics that tends to come up in other courses. Okay. So otherwise, everybody knows the drill, the scantrons, the pencils, the ID, all of that. So just be prepared for that on Wednesday. The sample questions have been put up on Blackboard for several days now. Excuse me? What did I write? To help? What did I say? Today's Wednesday. All right, let's be clear. On Monday, we're going to have the exam. As far as I know, that's correct. I'm planning on having it Monday. I don't know what you guys are going to do. I've given you 20 plus sample questions, about at least, probably right at 25% of the actual questions from the exam, and even again gave you a breakdown of approximately how many questions from each chapter. So you and the number of questions on the exam per chapters. The attempt is to weight them out. So topics that we cover in a lot of detail will be represented a little more extensively on the exam. Okay, so before we get into this, let's do our usual. So here's a question from the last lecture. And we'll let you work on that one. Well, this time we do have an answer E. Okay. All right. Since it's the first one, I'll go to 1 minute and 15 seconds on this one. All right. And the answer is what? Here we go again. Some little quirk in the software. Okay. Now, if you're looking at it and going, that's not what I answered last time, it's another one of those examples where you just reword the question and it's got a completely different answer. Okay, let's try, that's old news. Let's try something new. I'll do 1 minute and 15 seconds on this one also. let's see. This one tends to be all over the map. So, what do you think it is? Okay. Which, not too bad. 28% of you figured that one out. Okay. Hopefully the next time you get it right. All right. So we're moving into chapter 16. We started out last time and we talked about DNA binding proteins. And that's really at the heart of this whole chapter. And this is an area that is going to carry over into eukaryotic gene expression. It's this idea of a protein binding to a specific DNA sequence and affecting some type of an outcome. So in bacteria, you have this Operon model. What's really important here is this is unique to bacteria. You do not see this type of an organization in eukaryotic cells. The concept is pretty straightforward. The cell needs to carry out some metabolic process. Using the Lac Operon, which we'll talk about, the cell, if lactose is present, the cell needs to, that's a disaccharide, it needs to be able to first get that lactose from the outside world across the cell membrane into the cell, and then it has to break the disaccharide into the two monosaccharides, and then they can go off in their various pathways. And it really needs 3 different proteins to do that job. So the genes that code for those 3 proteins are all clustered right together, and they are the structural genes. The question is, the cell doesn't want to make those enzymes if it doesn't need it, so how is the whole process regulated? But the idea is that you cluster all of the genes necessary for that process, and put them together, and then flip it on and off under control of a single switch. This kind of coordinate gene expression happens in eukaryotic cells, but you don't see this Operon model. For example, we've talked about hemoglobin a number of times. Cells need to make equal amounts of alpha and beta globin polypeptides. You would think the simple thing would be put them together like in an Operon and just express them at the same time. But in fact, those genes are on different chromosomes. And so in eukaryotic cells, there's a different mechanism to regulate, to coordinately turn on expression of genes that are all needed at the same time. So with the Operon, we're really focused in a bacterial system. And there's going to be 2 parts to it. There's going to be a regulator protein. And the important part here, and what I was trying to get in that previous question, is it's not part of the Operon. And in genetic terminology, it's said to be transacting. It's a protein that can diffuse throughout the cell. So the regulator, the gene that codes for the regulator protein, could be on a plasmid and still carry out its function in controlling expression of an Operon that would be in the genomic DNA of the bacterial cell. The other part is the operator. That's a DNA sequence. And again, in a genetic terminology, it's said to be a cis-acting sequence. That means it has to be in the proper location next to the promoter. Okay, if you move the operator, you can move the regulator gene around in the cell, and the system will still function. Move the operator around, and it's not going to work. So the operator has to stay in the position where it is normally to function properly. So here's just sort of a generic Operon in bacteria. So we just simply have the gene that codes for the regulator protein, and that protein is going to bind to an operator. And that operator is going to be situated near or be somewhere very close to the promoter. And here's the structural genes. And whether that protein is bound to the operator or not is going to control whether transcription happens or not. All right. So we can really do this in 4 ways. We really only have 2 states. Either the protein is bound or the protein is not bound. And if the protein is bound, it can turn on gene expression. So if I say the system's turned on, I mean transcription is activated. Or the binding of the protein can turn it off. So that really gives us 4 possibilities, which we have here. Negative inducible, negative repressible, positive inducible, positive repressible. Looks crazy, but again, take a complicated process and break it down. The key to this is if the protein is bound, when the protein is bound to the operator sequence, if gene expression is off, we call it negative. If gene expression is on, If binding of the protein turns gene expression on, we call it positive. Inducible means that the default state is transcription is off, and we're going to have to do something to induce it or turn it on. And repressible simply means that the default state is that transcription is occurring unless we do something to repress it or shut it down. Those are the only four possibilities. And they're all controlled by whether or not this regulator protein binds to its operator sequence. Okay, so that's actually what I've said on this one. All right. There's actually going to be a third player in the game. So you got the regulator protein, you got the operator, and you're going to have an effector molecule. And that effector molecule is going to bind to that regulator protein, and in doing so, it's going to change the shape of that protein. It may change it in a way that now allows the protein to bind to the DNA, the operator, or it may change it in a way that doesn't allow it. And so we'll just show you examples of each of those cases. So here's a negative inducible operon. This is the Lac Operon. Well, actually, the Lac Operon has two of these in it. So the first part we'll talk about is the negative inducible. In this case, the regulator protein is made in an active form. By active, we simply mean it's capable of binding to the DNA. It is a negative system because binding of that regulator protein blocks transcription. So you see no transcription. Right? So it's an inducible system. The default state is for it to be off unless we do something that will now turn gene expression on. It's an inducible system because we need to add an inducer. In the Lac Operon, that's going to be lactose. In this case, in a more generic sense, it's going to be this precursor V. So this is what you're going to see in catabolic pathways, and that is the cell, these are going to be breakdown pathways. So the cell doesn't want to make the enzymes needed to break down the catabolite unless it's present. So for instance, it needs to break down lactose. Lactose would be the catabolite. So in this case, the inducer binds to this regulator protein, and in the catch-all term the biochemists like, there's an allosteric change, which simply means the shape of the protein changes. When it changes shape, it no longer fits on the operator sequence. So you induced it by changing the shape of the regulator protein. It can't bind to the operator, and now the RNA polymerase is free to transcribe the genes, and transcription is turned on. Okay. We can flip it around and make it a negative repressible system, in which case the protein is going to be made in an inactive form until the co-repressor binds to it. So it's the same concept, but in this case, we don't want to make the enzymes because we don't have precursor T present. I'm sorry. In this case, we're going the other. This is an anabolic pathway. So this is going to, I'll show you, it's going to be feedback inhibition. So the cell needs to make product U. That's the co-repressor. The default state is to be transcribing and translating these genes, making these enzymes, and converting precursor T through all these intermediates to product U until U gets to some level in the cell and the system says there's enough here. So let's stop making it. So the regulator is made in an inactive form. It doesn't bind to the operator, and transcription is on. That's the default state. Now, by feedback inhibition, when product U gets high enough, it binds to this inactive regulator protein, changes its shape, and now makes it in a shape that binds to the operator. And binding to the operator, since it's negative, binding to the operator blocks transcription. So this was what you would see in an anabolic pathways. When you get the final product to the level you need, then a feedback inhibits and shuts the system down. Okay? So those are the two negative ones. Very similar to it are going to be positive, except they're going to be different. The difference here is when the regulator protein binds, it's going to turn on transcription. So it's positive. So positive inducible means that regulator protein is made in an inactive form. So there's going to be no transcription because it can't bind, and if it can't bind, it doesn't turn on gene expression. When it binds the inducer, then it'll be able to bind to the DNA and turn on transcription. This is the second half of the Lac Operon of catabolite control that we'll talk about. So the normal, in this case, the defaults for positive inducible, the default state is transcription's off. The activator protein is inactive. It can't bind to the operator. If it can't bind, since it's positive, it means no transcription. Now, this would be in a anabolic pathway. Now, when you add the precursor, it's the activator, binds to the regulatory protein, changes shape, binds, and since it's positive, it turns on gene expression. And now you make the enzymes that you need to convert this precursor to the final product. And then the last one is just positive repressible. This will be just the opposite. Now the regulator protein is synthesized in an active form. That means it's going to be able to bind. If it can bind, will transcription be on or off? It'll be on. So the default state is it's expressing the genes until you do something to repress it. So the active activator is still the regulator protein. It's just now we're going to call it, instead of a repressor, we'll call it an activator. It's made in an active form, binds to the DNA, to the operator sequence, and transcription is activated. Until, and again, it's going to be another one of these anabolic pathways, feedback inhibition, once enough of this little diamond product is made, it'll bind to the activator protein, change its shape, it can't bind, and transcription is shut down. So it's just 4 different states, all controlled by whether or not this regulator protein binds to its target DNA sequence. Make sense? Well, yeah. Well, it's okay. All right. Let's see if you can decipher your way through this then. I think a minute should be enough on this one. Okay. Wait. All right, we'll go to 115. You hear cries from the crowd. No, stop, please. All right. So which one is it? Okay. If it says it's normally turned on, then it's going to be a repressible operon. You're going to have to do something to stop expression, and it's going to be turned off by a repressor. All right, let's try this one. What happened? Oh, I thought, oh, I gave him the answer again. Done that before. All right, looks like you're doing better with this one. Go to 1 minute on this one. Okay. All right. So what is this one? Okay. So inducible, if the system is normally off, you're going to have to induce it. You're going to have to do something to turn on expression. And so it's going to be kept off by a repressor. Yes? An activator. The inducible, in this case, like you talking about A?
Like when would you use activator?
Yeah, the activator, for the activator, the regulatory protein is going to, well, it's got to be let's call it an inducible activator. Let's call it in the positive system. You got a protein which is going to turn on gene expression, but it needs to interact with some other molecule to become activated. The Catabolized Repression of the Lac Operon's part is an example of that. Okay, so those are the four basic ways in which, so this is really all E. coli has available to it, are these four different states of being able to regulate gene expression. So we're going to use the Lac Operon because this is the model from E. coli that's just beaten to death, course after course after course, and students still going, I don't quite get it. But eventually you finally come off. If you understand this, they all work in a very similar way, except for the ones that don't, like attenuation. All right. So this is the Lac Operon. It was worked out way back in the 1960s by Francis Jacob and Jacques Monod, using, again, classical genetics. This was before genomics, bioinformatics, cloning. Just by using some very elegant genetics and very careful observations, they were able to work out this system. So here's the biology behind it. The cell needs to requires three proteins to be able to utilize lactose. We're not going to be making lactose, we're going to be utilizing it. So lactose is a disaccharide, so it's two sugars, and the first thing the cell has to do is break the bond between the disaccharides so that then it can shunt these monosaccharides off into the various pathways. However, it would be a waste of energy for the cell to make these three enzymes if there's no lactose in the medium. So the system is designed so that it'll get turned on, expression gets turned on when lactose is put in. So a modified, I'll show you in a minute, a slight modification of lactose is the inducer for this system. The three genes, the first is a permease. This is the product of the LAC-Y gene. The permease is a transmembrane protein, and the cell needs this to be able to transport the lactose from the extracellular environment into the cell. The second one is the major workhorse, beta-galactosidase. This is the enzyme that cleaves the lactose into the two monosaccharides of galactose and glucose. This is LAC-Z gene product in beta-galactosidase. You'll hear it oftentimes be called beta-gal. This is used extensively in molecular cloning technology, even in eukaryotic systems, because we have lots of different assays for this enzyme activity. Changing color, various other things. So as we get into the technology section of this, you'll see this beta-gal gene comes up in a number of cases. So it's been very widely used. The third one is interesting. So it's thiolgalactoside transacetylase. Everybody knows what that does, right? Well, the interesting thing is you can figure out the biochemistry of it, but at least the last time I went looking for information, even though this gene's been known since back in the 1960s, nobody really knows why it's needed. If you have a LAC-A mutant, the cells can't utilize lactose, but nobody's really worked out the biochemistry of why. Right? But those are the three structural genes. So those are the genes that are going to get transcribed and translated into proteins, and they're actually going to be doing a job in the cell. Now here's another very important point. This is a polycistronic mRNA. Cistron is an old term that means a gene. It really hasn't carried forward once people stopped kind of doing some bacteriophage genetics and such. But it still persists in this sense. What it means is the messenger RNA that's made from the operon encodes the information for multiple polypeptides. So in this case, there's 3 structural genes. There's going to be one messenger RNA that encodes all 3 polypeptides. Even for something like the his operon for histidine synthesis, there's about 11 enzymes involved. The polycistronic mRNA codes for all 11 enzymes. You don't see this in eukaryotic cells. Eukaryotic cells are monocistronic. Each mRNA codes for a single polypeptide. Okay, so here's what at least the 2 proteins that we know what's going on. So you have lactose out here in the extracellular environment. The permease is basically creates a channel through the cell membrane to allow the lactose to be transported into the cell. The lactose is this disaccharide, and so the beta-galactosidase actually has to do 2 jobs. One is it's going to break that bond and create the 2 monosaccharides, and it also makes a small modification to a few of the molecules of lactose to create allolactose. That's the repressor. I'm sorry. Yeah. This is the effector molecule. This is actually the inducer. This is the effector molecule that's going to bind to the repressor protein and cause the change in shape. Yes?
For like people with lactose intolerance, is that just an inability for like the lactose to break down lactose?
I'm going to guess so. I don't know that system in detail. But again, in a eukaryote, in a mammalian system, there could be several other enzymes involved. But yeah, most likely it's somewhere involved in this inability to break down the disaccharide. But that's really kind of a wild-ass guess on my part. All right. So the cell needs to know whether there's lactose in the medium or not. So if you grow cells in medium lacking lactose, and you measure the amount of each of these 3 proteins in the cells, the level is very, very low. However, if you add lactose, and this is an important point, and the medium does not contain glucose, then within a matter of minutes, the expression of those proteins goes up 1,000-fold. So there's a very strong induction of the expression of these genes within a matter of minutes after you add the lactose. All right. How this whole system is regulated really involved the discovery of 2 additional genes. The gene that codes for the regulator protein, which is called the LAC-I gene, and the operator sequence, the DNA binding site for that repressor protein, and that's called LAC-O. So there's really 5 genes that make up the whole Lac Operon. Only the 3 structural genes are actually transcribed, translated, and made into proteins. Is the regulator gene transcribed and translated? Got to make the protein. But remember, it can be located somewhere else. Actually, even normally on the chromosome, it's actually a little further away from the actual operon. Does the operator get transcribed? No. It's just the target site for the repressor protein to bind to. Okay. So here's the basic structure. So the regulator gene is upstream of the Lac Operon. And then here's LAC-P is the promoter. We'll talk about this in a little bit more. You'll see that LAC-O overlaps with that promoter. And then these are the 3 structural genes, LAC-Z, LAC-Y, and LAC-A. They actually get transcribed. Okay, so if we don't have any lactose, we make the active repressor protein. It binds to the operator and really prevents the RNA polymerase from being able to have access to and transcribe the structural genes. And so there's no transcription. Now, if we turn it around and we add lactose, then the lactose gets converted to allolactose. So this is the actual effector molecule. It's going to bind to the repressor protein. There's this allosteric change. Now the protein can't bind to the operator, and the RNA polymerase is free to transcribe and translate the genes. Sorry. So the whole system is just turned on and off based on whether or not the allolactose is there and what the effect is on the repressor protein. This just shows you at the nucleotide level. Here's that -10 and -35 sequences that you know everything about because you've been studying. Okay, this is where the RNA polymerase will bind. But the operator actually overlaps that region a little bit. So if you remember, the polymerase actually extends out onto the actual transcribed sequence by about 20 nucleotides. So if the repressor protein is bound right here, it's going to prevent the RNA polymerase from being able to initiate transcription. Okay. So it kind of begs this question of, it's kind of a chicken and egg situation. If the Lac Operon is repressed in the absence of lactose, and you need the permease to get the lactose into the cell, then how does the lactose ever get into the cell if there's no permease? And if there's no lactose in the cell, how can the whole system get turned on? Well, the answer is, if you think back, I remember I said initially there's a few molecules. These systems are not 100% repressed. So even in the absence of lactose, there's a small amount of these, a low level of transcription being made. So there's a small amount of the permease, a small amount of beta-galactosidase, and a small amount of the transferase. Let's see. And so that's really all that's needed is a small amount, just be able to get a small amount of the lactose in, and a small amount of the beta-galactosidase converts it into the allolactose. That's enough to bind to the repressor. The repressor shape changes, it comes off the operator, and then transcription accelerates. Okay. So does everybody see why if the system was 100% efficient, it wouldn't be efficient at all? It'd be completely broken. So it's got to be a little bit sloppy. This is IPTG, isopropylthiogalactoside. I put this in here because beta-galactosidase and modifications of this Lac Operon are used so extensively in molecular biology. For instance, I told you we make a recombinant, we make a vector that we put into E. coli to make a recombinant Drosophila protein. Well, the whole system is actually based on a modification of the Lac Operon. We could use lactose to induce the system to turn it on. The problem is the lactose gets metabolized. And once it gets metabolized, then the system will get repressed again. All IPTG is a non-metabolizable analog of lactose. So it does exactly what lactose does. You put it into the cells to turn on the Lac system, the Lac Operon. It binds to the repressor protein, changes its shape, but it can't be metabolized, so it's very stable. So it's just used experimentally as a substitute for lactose. Okay. So one of the things I'll suggest that you do to help learn how this system works is look through the textbook and do what Jacob and Monod did. They worked this all out by finding mutations. So you can ask yourself, what happens if I have a mutation that in the repressor protein so that now it's irreversibly bound to the operator. Then what happens? What would happen if, so you make a mutation, you have some mutation, and now the protein binds so tightly to the operator that even if you add lactose, it still remains bound. What would be the consequences of that kind of a mutation? The system on or off? Off. It just simply could not use lactose. If you had a mutation, let's say you had the opposite case. You had a mutation where the Lac repressor couldn't bind to the operator. Then what would happen? The system would be on constitutively or on all of the time. So you can just go through and just ask yourself, okay, what happens if I have a mutation in the operator sequence? What happens? And you begin to see how the system works and basically working it out the way these two investigators did. Okay? And there's several examples in the textbook and such. So I think it'll be more effective for you to go through that. Okay, so before we get into the second half of the Lac Operon, let's try this question and let you think about it for a minute. All right. I'll go to one. We'll go to 115. Okay. So what's the answer? Okay. Any questions? Before we go into the binding of the cap protein, just how many of you plan to look for a job after graduation? Let's put it this way. How many of you just plan to live with your parents until you are able to collect Social Security? Okay. The reason I say this, I was at a meeting with a pharmaceutical company up in Riviera Beach. And the interesting point was, and there's a lot of these companies are coming into everywhere from the Treasure Coast down through Miami. They're coming up. One of them I've worked with is called Boca Biolistics, except now they're down in Pompano, I think. The CEO was saying his main problem is he can't find people with the skills that he wants to hire. He's got one production of one of the products they're making is falling behind 3,100 hours a month because he could hire 15 people now to go in there. Now I know it's on the other area what he needs. For instance, he said he could hire 15 analytical chemists right now. He's taken on some students. Let's say now they're recent graduates. He said you can come in, with the right kind of background skills, and they have to do some training, but would be starting at probably about 45 to 50,000 with a bachelor's degree. He said within 2 years you'll probably be making either 70,000 a year or you'll be fired because they want you to be moving up. My point is that I would be looking around and seeing what are the jobs. I don't have a laundry list. I can't just tell you what everybody's looking for. But if you want to stay in the South Florida area, it was like when I was in California, nobody wanted to leave. And, and you're looking for good paying jobs. These companies are coming here. And what I would be doing is trying to figure out what are they looking for and what are the skill sets that they want to get in there. It's probably, now some of the jobs, they're at PhD levels, but it's like the analytical chemist. He said there's only a couple of PhD programs left in the country that are in analytical chemistry, but many of those jobs, very quickly, you'd be making 6 figures, in the job. It's not a bad environment to work in. So anyway, just something to think about if you're really getting tired of living with mom and dad. And believe me, mom and dad are getting tired of living with you. So, you had a question?
And did he say what skills specifically people were lacking?
Oh, he went through such a laundry list that, I mean, I'm sure on their websites and such, there's information there. You got to do your homework on these. But it sounded like, and he said other companies, he's talking to the CEOs of other companies in this area, and they're having the same problem. He does a lot of FDA trials work. So people for instance, we were talking to him about our professional science master's program, which is really, we kind of call it a mini MBA because it's half science, half business. He kind of perked up with that because there's a lot on the financial side. I don't know, he's just one part of it. It's not a big company, and he said just one division of it, they'll probably spend $30 million this year as part of the FDA trials and such. So there's actually significant amounts of money involved, and there's lots of opportunities. So if you're looking outside the conventional healthcare field, you want to look for something that pays quite well. It might require graduate level training, but what you're seeing is, one is you should be aware, you may not be aware. In the sciences, you have the advantage over, let's say, English majors in many places, that you actually get paid to go to graduate school. So, I mean, that's the advice I got, was have someone else pay for your graduate education. Okay, so you should be able to get a graduate degree in the sciences with very little, if any, additional debt. It's not, I was under a more ideal situation, but you can still get a big chunk of it covered, especially in terms of tuition reductions and that. And then you can step into some nice paying jobs with a lot of potential and places like that might even turn around and pay for you to go to graduate school. My daughter she's a graphics designer, so she's in a completely different area, but she's getting her master's degree, and the company's paying 75% of the graduate tuition and give her release time when she needs to go take the classes. So anyway, just something to think about. If you're comfortable at home, spoken with me. What the heck? Yes.
So would you recommend going to find out these specific companies?
I would be just looking for, for instance, the question is, where do you find this information? You got to go do your homework, and I really don't know. I can't say go to this website, but what you can easily start doing is scrounging around and looking for what the biotech companies are. These little companies are coming to this area. They're not going up around Jupiter necessarily. If you're a startup company and you need electron microscopy and high throughput genomics and such, yeah, you need the infrastructure of Max Planck and Scripps and such. But these other companies he's doing some manufacturing. Some of it is supplements, some of it's FDA regulated prescription drugs, a lot of quality control. That's why he needs analytical chemists and such. They don't necessarily going to be right across the street on Donald Ross Road. They're down in Deerfield and Boca, Pompano, these areas. But I'll bet if you go on their websites, you're going to see job openings, and they're going to be listing the skills that they're looking for, and those are the targets that you should be looking at.
I understood that, but how do you find out about the companies? What specific companies?
Then I would just be looking for just Google biotech companies here in South Florida, and you start, for instance, there's a required internship as part of the professional science master's program. We've got relationships with a number of companies now that I can put students in and do internships and such. But on the other hand, I just asked them, I said, go look around, see if there's a company that you'd hope they'd offer you a job. Because then we can make the entry and make the initial contacts and see what's going on. So I think, just a little bit of research and see what skills. The president of Akron Biotech here in Boca, I have to dig it out of my email. They just moved in some bigger facilities. And now that I'm thinking about it, she sent me an email with, there must have been 20 skill sets in there, and it was like, if you got students who can do this, let me know. Sort of, not all of them. They're just you're not going to have all of them, but you can see there's a wide range, and it's going to be chemistry, and it's going to be probably some math, but also some business, all of these sort of things. But the jobs are coming. It's coming to the areas. Okay. Back to what you need to know by Monday. If you don't get past this speed bump, you're not going anywhere. Okay. I know I'm the single biggest impediment to your career, right? Okay. The other side of the Lac Operon regulation is called catabolite repression. Here's the problem, or here's the challenge for the cells. They want to use lactose. They want to turn on the Lac Operon and express those genes and make those proteins when lactose is present. But if they have a choice, cells would prefer to use glucose instead of having to break down the disaccharide. So if both glucose and lactose are in the medium, they want to suppress the expression of the Lac Operon until the glucose is consumed. Because they can put, as it says here, saying because they can just shunt the glucose directly into the glycolytic pathway. So this is going to be called catabolite repression. The glucose is a catabolite. The presence of it represses the expression of the Lac Operon, even if lactose is present. So here's how it works. There's a protein called the CAP protein, for catabolite activator protein. It's a DNA binding protein, just like we've been looking at. Here, a little more precisely, it binds to a 22 nucleotide sequence. It's located actually just slightly upstream of the promoter. So the operator was really downstream of the promoter, pretty much positioned between the promoter and where transcription began. The binding site for the CAP protein is a little just at the other end of the promoter. So it's very close, just slightly upstream. Well, actually it does. It's just right at the 5' end of the promoter. Here's the whole key point. The binding, even if the Lac repressor is gone, so let's say there's allolactose present, and it binds the repressor, and the repressor is not bound to the operator, and the whole system is ready to go, it's actually a very poorly transcribed system. The RNA polymerase by itself in that situation, transcription occurs, but it's at a very, very slow rate. The binding of this CAP protein is going to dramatically enhance the efficiency of transcription. So without CAP, transcription can occur, but it's very, very slow. All right. So how does the cell know how much glucose is in the cell? Well, it gets it indirectly. It uses cyclic AMP, adenosine 3' 5' cyclic monophosphate. You've probably heard about it. It's oftentimes called a second messenger. It's a key molecule for transmitting information within the cell. What it's doing is the level of cyclic AMP is indirectly telling the system, if you want to put it that way, the level of glucose. And that's because the cyclic AMP levels are inversely proportional to glucose. The enzyme that adenyl cyclase, the enzyme that makes cyclic AMP, is inhibited by glucose. So at high concentrations of glucose, the adenyl cyclase is inhibited, cyclic AMP levels go down. When the glucose is gone, cyclic AMP levels go up. So there's an inverse relationship between cyclic AMP levels and glucose. All right, so that's what that says. So what's going to happen here is when you have low levels of glucose, you have high levels of cyclic AMP, and this CAP protein, this catabolite activator protein, can only bind to the DNA if cyclic AMP is bound to it. So you have to have this cyclic AMP CAP complex to now bind to this DNA sequence, and as it's showing you here, it's a little bit upstream or right at the 5' end of the promoter. When that gets bound, and assuming that the operator is free of any repressor protein, it enhances transcription, and you have a high rate of transcription, and you end up being able to convert lactose into the 2 monosaccharides. When glucose levels are high, cyclic AMP levels are going to be low. There's going to be very little cap cyclic AMP complex. The cap won't bind. And now, even though the operator is not blocked, transcription is going to be very slow because without the cap protein, it's a very inefficient transcription. So now the glucose levels, the cell can consume the glucose, but when the levels go down, cyclic AMP goes up, and now it can turn on the expression to utilize the lactose. So this lets E. coli preferentially use glucose before it begins to consume the lactose. So is this going to, what type is this going to be? Is this going to be positive or negative? It's going to be positive because the binding of the protein enhances transcription. And is it inducible or repressible? It's inducible because the default state is to be off unless you do something to turn it on or induce it. Okay? So CAP actually works with about 20 different operons in E. coli. It's not specific to the Lac Operon, but it does the same thing. Tying it back to what we talked about at the beginning, when you look at the three-dimensional structure of the CAP protein, it has this helix loop helix motif telling you it's a DNA binding protein. So what it's really doing is it binds the DNA, and just like we talked about before, it bends, it puts strain on the DNA, and the first step in initiation of transcription is that opening of the double helix to the single helix. That's the rate limiting step. So the binding of the cap protein facilitates the opening of that helix, and that enhances transcription. So it's the binding of the cap protein which bends the DNA, puts stress on the sequences at the promoter, and really facilitates this transition from closed to the open duplex structure. Okay? Any questions? Anything else you want to know about the Lac Operon? Okay. So now here's the trip operon. Hang on. This one's going to be extra credit. Because this is an example. So in the trip operon, it actually needs there's 5 polypeptides that make 3 enzymes. Does that make sense? Okay. Because 2 of those enzymes require 2 polypeptides. All right. Would these have a quaternary structure? Yes, because there's more than one. The one enzyme that functions by itself would not have a quaternary structure. Okay. This is a repressive. So the idea with the trip operon is this is now an anabolic pathway. So you're going to want to make tryptophan until you get to some set point, and then you want to shut the system down. So you want to be able to repress it. Well, this system takes advantage of the fact that in a bacterial cell, transcription and translation are linked together. So it's a process called attenuation. It's a little more efficient, a little more finesse, a little more fine-tuned than the system you have with the Lac Operon. But again, since it's unique to bacterial systems, I've just put it up here. I'm not going to talk about it in any detail, but I'll just tell you that at least one of the extra credit, I don't know, sometimes I do 2, I can't remember. At least one of the extra credit questions on the exam will be based on this attenuation system in the trip operon. But to my knowledge, there's nothing similar to it in eukaryotic cells because transcription and translation occur in different places within the cell. Okay. So that's what's on there. Okay, I just kind of noted it here. There's some topics at the end that are kind of interesting, but we're not going to go into them. Antisense RNA and ribose switches and such. Okay, so you'll see that at the end of the chapter, but it won't be on the exam. All right. So let's try this question. All right, I think 1 minute should work for this one. Okay. And what's the answer? Okay. Any questions? So this is the formal end of the material for exam 3. What I want to do is probably take 15 or 20 minutes to start into the next chapter just to really keep us on track with the syllabus. The next chapter is eukaryotic gene regulation. Start to see where the big differences are and where the smaller similarities occur. But it will begin the material for exam 4. So that may be the cue to sort of shut down at the moment and stop paying attention, but that's your choice. And then around, let's say around 20 after, something like that, or I'll find a good stopping point. Then the rest of the period that we have left will be for any questions related to the exam, or if you have no questions, then that'll be it for the day. So moving into chapter 17 is what's really more important to current research. Well, I wouldn't say that. There's still a lot of important research being done in microbiology because of drug resistance and such. But the main emphasis, especially in terms of diseases and such, all looks at regulation in eukaryotic cells. Okay. One of the areas where you see evolutionarily the carryover from prokaryotes to eukaryotes is this theme of DNA binding proteins. However, as we've talked about before, eukaryotic cells, especially multicellular organisms, need more fine control over this level of gene expression. So not surprisingly, there's going to be a lot more players in the game. All right. And the other is that you do not see this operon type of organization in eukaryotic cells. And again, you don't have polycistronic mRNA in eukaryotic cells. Each transcription unit, each messenger RNA in a eukaryotic cell codes for a single polypeptide. Okay. So that's really what this is saying is, so that's why we use this term monocistronic. It's just about the only situation I can think of where currently the term cistron is used, but really means gene. So you have one gene being transcribed in eukaryotic cells as opposed to this polycistronic mRNA that we find in prokaryotic cells. The other is we have this chromatin problem. We have the higher levels of organization of chromatin. We've already talked about, so some of these slides are actually a review from the previous discussions, except now we're thinking about in terms of gene regulation is we have these nucleosomes. We'll see that the nucleosomes have to be moved out of the way. The chromatin, if it's in a very tight configuration, has to be relaxed and go from this heterochromatin to euchromatin. So there's many more levels of events that have to happen before the gene can be transcribed. Okay. So, I know that song. You know, I think one of the depressing parts of getting older is I'm an old school rock and roll music person, which was the music I grew up with, and now it's on the oldies station. Although my daughter, who's probably just a couple of years older than you guys, it's like, I listen to the music she grew up with, and I said, this is so depressing. And you're old like me, you're oldies, you just have nothing good to listen to. I tried to teach, I wanted her to appreciate classical music like the Doors and such, but anyway. I digress. I do that sometimes. Okay. Here's kind of a tricky point. We said there's no sigma factor in eukaryotic cells. The sigma factor is there to bring the RNA polymerase complex in prokaryotes to the proper site on the DNA so that transcription initiates at the proper nucleotide, at the +1 site. In eukaryotic cells, that RNA, that basal transcription apparatus, that RNA polymerase with all of its ancillary factors, all the TF2 factors, can in fact find its own promoter. So it doesn't need a sigma factor to find it, but it can't be activated unless you have these other ancillary proteins involved. And that's really what these transcriptional activators are. These are those proteins that bind to the regulatory elements telling you it's a DNA sequence, and then communicating through that mediator, then they're able to activate transcription. We talk a lot about transcriptional activators. Just to be clear, there are transcriptional repressors in eukaryotic cells, so the binding in this case, they would repress transcription, but they're in the clear minority. The vast majority of these the vast majority of these are transcriptional activators. Okay. So part of this is to go back to something we talked about before, and I will just talk about in just a little more detail, and that is you have this chromatin. So unlike bacterial cells, you got the DNA wrapped around the outside of this histone-based nucleosome core, and then it's all compressed and supercoiled on top of itself and put into higher orders of coiling. If it's too tight, then that basal transcription apparatus can't get to the promoter site, and the whole system has to be relaxed a little bit. Okay, so we got to make these changes in chromatin organization. So that's what it's trying to show here. The first part is they call it chromatin remodeling, but it's literally having to reposition the nucleosomes so that the DNA sequence is available. So that's what's happening here. So these chromatin remodeling proteins are literally moving the nucleosome positions so that now they've exposed a binding site for one of the transcriptional activators. So that's the first part, is to get the nucleosomes out of the way, and so that you have a segment of the DNA available for transcription to begin. And then what you're seeing here, this RNA polymerase, and this is really that whole basal transcription apparatus now has access to the DNA. So this is called nucleosome, well, it's repositioning of the nucleosomes. And one of the jobs done by this family of proteins called chromatin remodeling proteins. So it's kind of something you've seen before. Again, these DNA probes were used. The idea being, let's look for the sites that are most accessible to this nuclease. We're using this nuclease as a tool to see what part of the DNA is most accessible. Remember, the idea is, well, you know all this because you've been studying. So the idea is that if the protein is tightly associated with the DNA, then the DNAs can or cannot degrade it. It can't. So what you're looking at is the hypersensitivity means if I had just a little snit of the DNA polymerase, which DNA sequences get cut first? And that'll be the DNA that's most accessible. The chromatin will be in its loosest conformation, and it won't be associated with nucleosomes. So these are these DNase 1 hypersensitivity. We talked about this with the globin gene expression in the chick erythrocytes. You can do this in a more global sense, looking at a larger number of genes. And what you find is, just in general, genes that are going to be transcribed have a hypersensitive site about 1,000 base pairs upstream of the promoter. So what you're seeing is whenever the genes are going to become transcriptionally active, that DNA is in this looser conformation and it's readily accessible to the nuclease. Okay? So it's the same thing that we saw before, except now just sort of thought about on a much broader scale. Now the other thing we talked about, and I'll show you just in a little more detail here, was how tightly is the DNA being held to the histones. Now the histones are rich in lysine and what else? Arginine? Okay, you're getting there. All right, because they're basic proteins, which means they're positively charged at a physiological pH. So they're positively charged, they neutralize the negative charge on the DNA, and therefore the histones are binding more tightly to the DNA, and it's going to be harder for those chromatin remodeling proteins to move that nucleosome around onto another segment of DNA. So to help facilitate this relaxing and changing of the DNA conformation, we'd like to neutralize that positive charge, and that's where histone acetylation comes in. So this is an acetyl group. I'm not going to ask you to memorize it. Basically, it's going to be added onto the amine group that's positively charged on the lysine and the arginine, and in doing so, would this make it into a polar molecule or an aliphatic molecule? Right. But would this entity probably be hydrophilic or hydrophobic? It's going to be hydrophilic. It's just going to be uncharged. So it's still going to it's going to be soluble. It's going to be hydrophilic. It's just you no longer have the positive charge. Now the histones can't bind, interact with the DNA as tightly, and so the whole system kind of loosens up a little bit. So this histone acetylation is going to be associated with regions of the chromatin where the genes are getting ready to be transcribed. Okay, so that's what I was trying to show you in this cartoon. So here's the positively charged tails on the histones. They're tightly bound to the DNA. Because the protein is really so closely associated with the DNA, it's going to be resistant to the DNase. So is this going to create a hypersensitive site? Well, you got to read down here. It says DNase hypersensitive. Okay. So when you acetylate it, so when the acetyltransferases acetylate the histones, you take away the positive charge. As they're trying to show you here, those histone proteins aren't bound to the DNA as tightly. Now the DNA is a little looser on the surface, more accessible to the DNase, and creates these DNase hypersensitive sites. What it's telling you is this histone acetylation is being done in preparation for transcribing the genes that are located on that DNA. Okay. Just an example that you can read to see a little bit more of how acetylation and deacetylation regulates gene expression. And for the three of you who are actually interested in plants, I don't know, I always talk to Dr. Zhang. It's like, why don't more students want to know about plants? He's our genetic, he does molecular genetics with plants. It's cool. He's got the DNA gun, which you can do transformation of genes with, or do transformation of plants. Anyway, but it's an example. I won't go into the details. It's just for you to be able to get another idea of how this acetylation, deacetylation is involved in gene regulation. The other thing that comes in is what we've seen before too, is DNA methylation. This type of chemical modification of DNA is turning out to have many, many, many functions in terms of DNA metabolism. And what you find is, if in eukaryotes, as I've shown you in a previous slide, if the DNA is methylated, it's most likely going to be at position 5 on the cytosine. So you're going to have 5-methyl-C. This is on the base, not on the sugar. So this methylation occurs on the C residues. In bacteria, where does it occur? Primarily on 7-methyladenines. Okay. Which is actually the gene that codes for that methylase is called the DAM methylase, so the deoxyadenosine methylase. But I think it was actually found by a graduate student who finally went, damn, I'm actually going to graduate. Finally found it. All right. What you find is heavy areas of methylation are associated with areas that are not being transcribed. Okay? So the heavily methylated areas have repressed areas of transcription. So would you find this in the heterochromatin or in the euchromatin? Which one would be more heavily methylated? The heterochromatin, because that's where you're basically shutting down gene expression. Okay. The bar body in female cells of mammals is heavily methylated because there's virtually no transcription going on. In unmethylated areas, that's where the areas of active transcription are. So we can begin. This has been looked at for a wide range of genes, and it's very consistent. Heavy levels of methylation, low levels of transcription. High levels of methylation, low levels of transcription. Very little methylation, these genes are going to be actively transcribed. When you find it, they occur in so-called CPG islands. All that means is the P just represents the phosphodiester bond. So in most cases, when the C is methylated, it's usually followed by a G, and that's all they mean by CPG islands. Okay. Okay, so in here, as it says, you see, for instance, you see a lot of these CPG islands in inactive areas such as the bar bodies. Okay. All right. So let's try this question, and then we'll start to think about whether we might be at a stopping point. The other is, okay, I'll just say, let me get this started. I still have a number of unregistered iClickers for any number of reasons. So since we'll probably be quitting here in a few minutes and we got time, I'll put up the rolling registration again. If you have a few minutes, check the list and see whether your name is on it or not. All right. We go to 1 minute and 15 seconds on this one. Okay. But yeah, I think this would be sort of a typical exam type question. All right. So what's the answer? All right. So let's look at it. Okay, first of all, you're looking at the promoter for the insulin gene in chromatin isolated from brain tissue. Would you expect insulin to be expressed in brain tissue? No. Okay. So if the gene is going to be inactive, it's most likely going to be resistant to DNase, and it's going to have extensive DNA methylation. Now, if we did the same experiment, but we're looking at chromatin isolated from pancreatic cells, then is there a correct answer up there for that? That would it be A? No, because you still got the methylation. It would be B, the reverse. You'd be hypersensitive, suggesting the DNA is in a more open. What did I say?
It would be.
Oh, okay, this is. All right. Oh, hyper. Okay, I'm trying to read up the screen. Okay. So you're pointing the right one. So it would be hypersensitive to the DNA because the chromatin is in a relaxed configuration. The nucleosomes have been moved out of the way. The DNA is accessible to the nuclease, and you would have low levels of methylation. Okay.